Bé, bona tarda, good evening. Estem molt satisfets. We are very happy to have with us Professor Reinilde Beugelers to give the last talk in this annual cycle, Dijous de SECCT i Tecnologia. So, Reinilde, welcome to the Institut of Catalan Studies, Institut d'Estudis Catalans. Even though we are separated from a few thousands of kilometers, but thanks to technologies, we can get connected and listen your talk. I will now make a quick presentation of your profile, and then I will pass the floor to you. I will do this in català. Bé, com dèiem, bona tarda a tothom. Moltes mercès per ser amb nosaltres en aquesta sessió que per diferents raons doncs, l'hem hagut de fer així en línia. Reinilde Beugelers és catedràtica de la Universitat Catòlica de Lovaina, a Bèlgica, dins del Departament de Gestió, Estratègia i Innovació. És Senior Fellow de la Fàbrica d'Idees Bruegel des del 2009 i Senior Non-Resident Fellow del Peterson Institute for International Economics des del 2020. També és Research Fellow del Centre for Economic Policy Research, membre de la Royal Flemish Academy of Belgium for Sciences i de l'Acadèmia Europea. Del 2004 al 2008 va gaudir d'una llicència acadèmica com a assessora de la Comissió Europea, concretament del Bureau of European Policy Advisors, BEPA o BIPA. Ha treballat al Consell Científic del Consell Europeu de la Recerca, ERC, prou conegut, del 2012 al 2018, i també a l'EU RISE Expert Group. RISE vol dir Research Innovation and Science Policy Experts, i per tant ella ha treballat en aquest grup com a assessora del comissari de recerca. És membre de VARIO. VARIO és el Flemish Advisory Council for Innovation and Enterprise, un grup d'experts que assessora el ministre flamenc per a la innovació. Actualment és membre del Board of Reviewing Editors de la revista Science i co-IP de ciència de la Science Funding Initiative del National Bureau of Economic Research dels Estats Units. Amb la recerca centrada en els camps de l'organització industrial, l'economia i l'estratègia internacionals i la innovació i la ciència, és autora de nombroses publicacions altament citades en revistes internacionals de primer nivell. Algunes de les temàtiques específiques tractades recentment fan referència a novetats en el desenvolupament tecnològic, transferència internacional de tecnologia a través d'empreses multinacionals, cadenes de valor en innovació global, empreses innovadores joves, innovació per al canvi climàtic, relacions indústria-ciència i el seu impacte en la productivitat innovadora de les empreses, avaluació de polítiques de recerca i innovació, explicació de la producció científica, mobilitat internacional d'investigadors i recerca científica nova. Com veieu, amb aquesta trajectòria que hem exposat aquí resumidament, doncs seria molt difícil o gairebé impossible tenir una persona que ens pugui parlar amb més coneixement de causa del suport públic per a recerca de risc, i ella ja ha afegit un interrogant, errant l'objectiu. So, Reinilde, thank you so much for sharing your time and your knowledge with us today. I just presented very briefly your profile. And when I finalized my little talk in Catalan, I said that we can hardly have a better person, a better expert to talk us about public support for risky research, missing the target, which is the title of your talk. The floor is yours. Thank you so much.
Yeah, I'm going to share my screen. And in the meantime, I want to say that I'm very grateful for getting the opportunity to present on this topic, which is really very close to my heart here. And I hope to convince you of the importance of this topic here also for you. So you see the slides?
Yes, we see them.
Yeah, okay, so I can go ahead. So what I want to talk about is public support for risky research. This is a joint work that I've been recently working on also together with some of my good co-authors, Paul Stefan from the University of Georgia, Cheng Wang at the University of Leiden, and Chiara Frinzoni from the Politecnico of Milan. First, let me try to convince you why I think we should care about risky research and whether or not we are sufficiently supporting risky research here. So why should we or would we care about risky research? So risky novel research is something that's really necessary for pushing the scientific frontier to move here. Incremental research will only get you so far. And if you really want to get the scientific breakthroughs, the high gain, those are usually associated with high risk. So we don't obviously care about risk per se here, but if high risk is very associated with high gain, if we want a high gain, a scientific breakthrough, we will have to make sure we also accommodate the high risk component of that here. So novel risky research is more likely to become a big hit, but also more likely to fail here. And that's something that we have also already illustrated before here. Novel science is, of course, only one dimension of riskiness here, but that already illustrates that this novel science is likely to get to more breakthroughs, but at the cost of high risk, and therefore also more likely to fail here. So if we want the high breakthroughs in science, we need to take the risk, but it actually goes beyond that here. So also if you look beyond the scientific world, we also know that high gain and high risk research is important as a source for technology development here. We have also by now quite some research that shows that it's really the highly cited breakthrough type of science, which is significantly more likely also to be cited in patents here, so serve as prior art also for stimulating technology development as witnessed in patents here. And it's not just only that highly cited science is more likely to be cited in patents, but also in the most valued patents as well here. And on top of that, it's not just the highly cited breakthroughs here, but particularly if those breakthroughs come from science that's really more novel and has really this higher risk research here. It's not only that it will become cited much more likely in patents here, but also to reach completely new technology fields here that were previously non-impacted by the scientific discipline here, and are also way more likely to reach new technologies that are themselves also highly novel and highly risky as well here. So it's not just only that this high risk type of research leads to high gain and therefore more technology impact here, it also leads to new technological areas which also come with a higher risk here, but also with more new technology breakthroughs here. So I hope I've convinced you that we should care about this high risk here, not because of the risk, but because of if we want these high breakthroughs, both for science and for the technology impact, we should look at whether we are sufficiently supporting the risky component of that high potential high gain research here. And then, of course, there is the issue of, are we sufficiently supporting this risky research here? And then particularly, is there enough public funding support for the risky component of research here? If I haven't convinced you yet of this, the case of mRNA vaccines is actually a very nice one to make the case why we should care about risky research and why we should really also make sure that it is sufficiently funded or not. So the case of mRNA vaccines, of course, illustrates, if we go to the COVID-19 vaccine case here, the speed with which these vaccines were developed and how and their efficacy really underscore the importance of support for science, early science and research here. And particularly within the COVID-19 vaccines here, the most successful ones, certainly ex post, were the ones based on the mRNA technology here. But that was really the first human vaccine based on the technology here. So it was really the first time we actually saw the application of the mRNA technology for vaccines here. And in that respect, this was really a case of a high breakthrough that was very relevant and very important also for the technology developments for vaccines here. The advantages were the fact that they were very fast, safe, and efficacious, but also very quickly to adapt to new variants of the virus here, and also very importantly because they were because they were synthetic, they also allowed for the production of mRNA-based vaccines to be much more quickly scalable here and modularly produced here compared to the existing technologies which were much more biology determined and therefore much more difficult to scale up here. So mRNA ex post really turned out to be a very promising and transformative technology here that really very successfully survived its first big test here. And it was not just only for COVID-19 vaccines. Now that it actually was successful, it's also being further developed and is a promise for also future vaccines, but also future medicine in general, particularly also including for cancer and other. Yet the question is, I mean, was this expected? Was this in the making, or were we really lucky this time here? So if you look at the case, at the history of the research that underpinned this mRNA technology here, that goes way back in time here. So the discovery of mRNA goes back to 1961 here. A very next important step was how to synthesize mRNA here. That took quite a long time, but in 84 here. And then another important step in the whole process was in the 90s when they actually discovered that this synthetic mRNA, that actually the body cells could use and create proteins from this messenger mRNA delivered to them here. And that, of course, opened up the possibility of using mRNA and synthetic mRNA for drug development here. But there was a very big problem that needed to be solved, and that was that this synthetic mRNA, when injected into humans or animals, that there was an immune response effect here. And that actually caused a lot of delays and a winter in mRNA research here. And basically, actually, in the scientific community, this was seen as a non-go here, in the sense that there was this critical problem, and nobody actually believed or was really very passionate about what this research trajectory could actually bring here. Fortunately, there were always these stubborn researchers here, and in this case, we did have a few stubborn researchers. One important one was Katalin Karikó, a young Hungarian biochemist, and she was determined to find a way to make it work, the synthetic mRNA here. She was really focusing initially on more strokes and cerebral diseases here. And she tried and she tried. She had a position at the University of Pennsylvania, a soft money position, for which she had to submit grants to the NIH here, grant applications to the NIH in order to fund her position. She submitted lots of them here, but never got any funding here. And she sent us her referee reports that she got, and they were all about, this is very interesting, but it's way too risky. There is no proof that anything of this will work here, so we think it's too risky and we will not fund it here. And because she couldn't get funding at the NIH here, also the University of Pennsylvania was actually no longer willing to support her and put her actually on a very fluffy postdoc kind of position here. This was the position that she was in here, but nevertheless, she didn't give up here. She stayed at the University of Pennsylvania. And then there was this fortunate photocopy machine moment here in 1997, where she met Drew Weissman, who was an immunologist at the University of Pennsylvania, who had just returned from NIH to work at the University of Pennsylvania. He was also interested in mRNA technology to produce vaccines here, because that was his background here. And the combination of their passion both for mRNA technology and making it work, and the combination of the biochemistry expertise of Karikó with the immunology expertise of Weissman, they started working together. Of course, they had no funding, but Drew Weissman could actually use some of the funding he had from other projects to on the side fund this joint research here. And they came up with a big breakthrough when they understood what was causing the immune reaction here, the protein that was causing this, uridine. And they also identified that if they replaced the uridine with a pseudo-uridine, that they could actually evade the immune reaction of the body here. And that, of course, was a big breakthrough here that opened the way for using mRNA technology, synthetic mRNA technology, for drug development here. They managed to publish their results in the journal Immunity. They first tried with several top journals like Science and Nature here, but got rejected here. Immunity, it's also a good journal, but it's a very field-specific journal here. Initially, they didn't get many citations, so it took quite some while. Now it's a highly cited paper, but certainly in the first years, they only got very few citations here. So that's a bit the story in terms of science. If you go a bit further, so after that, after their discovery, Carrico still continued to have problems getting funding when she applied for follow-up research here. But also Weissman was a bit more successful because he had a bit more reputation already, but even he had a lot of problems here because still the scientific community still was not really convinced yet of the mRNA technology here, so even initially. The University of Pennsylvania, so the discovery had to be disclosed to them because of the Bayh-Dole Act here, and applied for patents. And the University of Pennsylvania was not that particularly interested in license actually the resulting patents exclusively to another outside company, CellScript. And actually what was really interesting is that Carrico and Weissman started their own startup here in order to go further with drug development here. But their own startup didn't even manage to secure a licensing agreement with the company that owned the patent here. And so they had to give up their own startup here. Others like Derek Rossi, who's by now very well known because he co-founded Moderna, he also took up the founding of Carrico and Weissman here and started to use that to develop drugs, but more for skin cells and for cancer treatments here. Carrico eventually left the university, at least to some extent, and she became she became the vice president research at BioNTech. By now, we know it's Moderna and BioNTech, which were really the driving forces behind the mRNA-based vaccine for COVID-19 here, and also are very successful now, thanks to their success for COVID-19, to further continue to use the mRNA technology also for other drugs, including for cancer treatments here. So ex post, this was a success, but if you go through the story here, you could also say that we were a bit lucky to get it here, and we could have missed this here. So of course we have no counterfactual here, but we have no way to know what would have happened if Carrico's early applications would not have been turned down, but that she would have been funded. Then maybe by already before we got the mRNA-based vaccine for COVID-19, we would have perhaps already had other drugs based on mRNA, for instance, for the swine flu in 2009 here. But of course, we don't know here. On the other side, perhaps actually, if it wouldn't have been for the photocopy moment with Weissman, she would have given up and we might have never had the mRNA technology developed, also not for COVID-19 here. So what we only know is that her early proposals were not funded. The university also moved her out of her soft money faculty position here. Now, that could have been simply because she was bad in writing proposals here. She wasn't convincing enough to the referees here. So it could be just a quality issue of her applications and of her way of formulating her research here. But it could also reflect risk aversion on the part of review panels here that considered the area as too risky to be funded here. So in any case, I think the example does show that there is some kind of concern that early funding of, particularly the early funding of the designer mRNA research, now obviously a success here and a big breakthrough here, and very important also for follow-up development of drugs here, that this initial early funding was definitely difficult, and it could be related to this risk bias here. So all this to hopefully identify that this is an important issue to look at. So why we should care about risky research here, but also definitely why we should care about, is it sufficiently funded or not? Because for funding risky research, there is really a very important role for public funding and for government support for that here. Why? Because there is, you could say that actually the science system has a bit of a problem dealing with risk here, because risk, given the high risk, the large variance of outcome, researchers will typically be less inclined to take risky research here, avoiding this potential for failure here, and go for the more easier, incremental, lower risk type of research here. So the individual researcher may not have the optimal incentives to take on the high risk here, and that's exactly why the public support system should provide them with enough protected space, what's in the literature called, give them truly legendary freedom here, such that they get the scope and the incentives to go for the higher risk, higher variance type of outcomes here. So that's a fundamental reason, also already identified since long, including by Kenneth Arrow, Nobel Prize winner. The fundamental reason why we need government support of public research is actually because it's the government that really needs to promote this risk taking here, is able to take that risk here and induce individual and provide the resources such that individual researchers are willing to take a higher risk position here. Then the question is government and public support sufficiently doing that, taking that risk here? And again, that's a debate that we have already in the scientific community and beyond for quite some time here. A perception that actually that's not maybe what the government is doing sufficiently here. So there is this perception and a discussion that the appetite of public agencies to support risky research is actually, it's not high, and may actually be on the decline here, and that they are increasingly risk averse here. Just one quote here to illustrate that a little bit, but it's an ongoing discussion here where we can also discuss this later on. A quote from James Rothman, who was a Nobel winner in 2013 for medicine here. And so when he was interviewed, because of his Nobel Prize winning, what he said was actually, initially I really had a lot of failure here, and I was really happy that at that time they were willing to support me, even if I was failing a lot and couldn't really immediately deliver here. And he was saying that he had the impression with the younger generation that he saw that this was way less likely to be the case here. So given that he's a Nobel Prize winner, I think it comes from a good source here. So risk aversion in funding scientific research, there is a lot of discussion on that, but the question is, do we have any more systematic evidence on that beyond the casual that it truly exists? And even more importantly, if it exists, what are the sources behind this potential risk aversion? And are there any possible remedies here that we could actually suggest, such that we at least avoid missing the targets of risky research, and therefore the potential breakthroughs that would come from that here? That's something we also already discussed more generally in another contribution together with Chiara Franzoni and Paula Stefan. So if we want to do that more systematic evidence here, we first have to identify what risk is and how are we going to measure that. There is already big discussion on that here, but just to land a bit on that discussion, to be able to measure it, the way we use the term risk is referring to the uncertainty on the outcomes of research here. And risk means that the outcomes vary in the spectrum of gains here, that the potential of exceptional results is high, but at the same time also the potential for no results here. So risk is therefore uncertainty in outcomes here, high potential for for extreme positive outcomes, but also for the extreme negative outcomes here. That's how we would define riskiness. So looking at the outcomes and this high dispersion of outcomes here. Now, it's very hard to measure that dispersion of likely outcomes here. What most of the research that looks at this does is it tries to identify what type of risk would have that outcome, uncertainty and outcomes here. And then let's study those particular types of research here. And one particular way one particular type of research that has these features is novel research here. And with novel research, we mean research that actually uses building blocks that deviates from previous research here. So it is novel, what deviates from what was already done in terms of research before. That's what we would identify as novel. And if that really has these features of leading to this high uncertainty in outcomes, then although we're not measuring risky research, we're measuring one type of research that has this higher risk here and so it would be interesting to look at whether there would be a bias against that novel research, just realizing that that just may be one dimension of risk, but there could also be other dimensions as well. So just to convince you that this novelty measure is something that really captures risk here and therefore is relevant to look at in terms of whether there is a bias against it or not, is something that we looked at previously in another contribution together with Cheng Wang. So there we measured so we take a combinatorial measure approach here. So we assume that building blocks are always combinations of building blocks here, and when there is a new combination of building blocks, that's combinatorial novelty that we want to identify here, and we measure that by whether a paper makes new combinations of reference journals here. The reference journals are the building blocks here, and if you make new combinations of these reference journals that have never been made before, that publication is then classified as novel here. And the interesting thing was that this novelty really, all these publications that have this novel classification here, first of all, they're very rare, not many of those. So it's only about 5 to 10% of publications that have this characteristic here. But more importantly, if we look at their citation performance here, so on average, they have a higher mean citation performance, but most importantly, it comes with a higher variance here. So these kind of novel papers are much more likely to score as a breakthrough, but at the same time also as a higher likelihood to be actually missed and to be not cited at all here. And so that really enforced us to think of, indeed, novelty seems to have this risky characteristic here. Furthermore, although there was a higher likelihood that high impact would occur, it takes a much longer time for that higher impact to realize here. So in contrast with other high impacts, which comes much faster here, novelty, when it becomes a high impact, takes a much longer time here. Typically, it takes about 3 years, but novel research needs at least 5 years before you actually get that high impact. And also the high impact actually comes from, certainly initially, from outside your own field here. So it really, again, is this riskiness that's very often embedded in existing communities here, and the impact takes a longer time. And you first convince, it's easier to convince others outside your field than your own field here. And part of the reason why the impact also takes longer is because this is very often published in journal, not in the top journals here, but journals with lower impact factor here, and that partly explains why it takes longer to get the impact here. And so that also means that if we use our classic bibliometric indicators, which use journal impact factors, these journal impact factors we know are based on quite short citation windows, typically 3 years. So they may actually bias against this novel research here, because their impact scores are usually take much longer than 3 years here. So the classic bibliometric indicators may not be picking up the novelty and the potential for the impacts from novel research here. So just to illustrate very quickly, this higher impact takes a longer time here. So here you see the highly novel papers, and the likelihood that they would actually be top-cited. And you see that with over time, initially they are less likely to become top-cited here, but it's only after at least 3 years that they outperform other research in terms of likelihood of being top-cited. So it illustrates the time it takes for high impact here. And on this, you see that the high impact actually comes from not your home field, but actually it's really in the foreign fields that you outperform, that this novel research outperforms other research in terms of high impact here. So we were a bit convinced that indeed this novelty measure is something we could use actually to assess any risky research and whether there would be any bias against risky research by looking at biases against novel research here. We were not the only ones to do this here. Others also looked at this. For instance, Kenneth Boudreau from Harvard conducted an experiment on peer review, and he found that novel research proposals that he measured with keywords that were new and previously not used, that those research proposals actually received much more negative evaluations during peer review compared to others here. And what we did is we used our novelty measure that I just explained above to look at the European Research Council and to see whether there would be any bias against the applications for novel research at the European Research Council here. Now, so why did we think that the European Research Council was a good case to look at here? Well, first of all, the European Research Council in its initial mission statement was really very clear that it wanted to go for the high gain, high risk type of research here, which means that it definitely should have no bias against risk here. Of course, it was not just purely going for risk, it was going for the high gain, high risk here, but it should definitely not be biased against risk taking here. So if you read its initial mission statement, it said very clearly, of course, first of all, scientific excellence is the sole selection criteria here, but really looking at the high gain, high risk pioneering proposals that really introduce unconventional, innovative approaches and to bring that research that will bring new and unpredictable discoveries here. So clearly, if you read this mission and you believe these mission statements, it should have no bias against novelty here. So that made it an ideal case to look at, is this really true or not? And second reason was because the ERC, as you all know, was really also very special because it has had really very big sized grants here, which could go up to €2.5 million, and also for a long period, for 5 years, grants for bottom-up proposals. And that large amount of money and long time really should be able to create enough protective space for researchers to be able to go into the more risky type of research here, because the money that they had available and the time period that they had available for working with that money should allow them to take more risk here. So in that respect, we were also expecting for a high treatment effect, that the ones that received the ERC grant should also have less of or at least should have removed their bias against risk themselves here, and should be more likely to take risk compared to the ones that would not have gotten the grant here. Before I show you a bit the results of what we found here, also important to identify is how the ERC operates in terms of grant evaluation. So the evaluation of the grant applications is by peer review panels, where the scholars in these panels are selected on the basis of their top excellence at world level here. So this is supposed to be top experts at world level. Also, in the second stage, there are remote referees, also again selected as top researchers here. It's a two-stage selection. In the first stage, it's the CV and the synopsis of the proposal. In the second stage, it's the full proposal. This might matter because in the first stage, there is way more emphasis on the CV and only a synopsis of the proposal. And of course, these are way larger numbers than in the second stage. So we would expect that in light of the potential bias of the bibliometric indicators, which are more likely to be used in the first stage here, that if there would be a bias, it would be way more likely in the first stage than in the second stage here. Again, the peer review panels are independent to select the proposals that they think are the best to fund, and they have full independence on this. But they are asked by the ERC to really not to shy away from risky research here, and really push for the frontier and for the groundbreaking nature here. The ERC does not provide or ask for bibliometric indicators, but what was often observed is that the PIs either self-report these bibliometric indicators in their applications here, or panel members, and particularly in the first stage, self-search for bibliometric indicators if they would be not provided. So overall, definitely also, as in many other selection processes, these bibliometric indicators do play a role. So how did we go about to analyze whether the ERC indeed is potentially biasing against risky research or novel research here? So we looked at two questions. One was, what does the ERC select? So that's the ex ante question. So what we did is we compared the ones that got the grant versus the ones that did not, the rejected ERC applicants. And we looked at the pre-grant publications of these granted versus failed applicants, and whether in these pre-grant publications, the ones that had more novel publications were biased against compared to the others. Of course, also checking for the excellence, the big impact as well here. So that was one thing. So do we see that that those applicants that have a record of more novel research publications, are they biased against compared to the others, all else equal? And the second thing we looked at was the ex post analysis here. What's the impact of ERC funding? We checked actually the grantees that got funding here on their post-grant publications, and we compared them with counterfactual similar grantees but without ERC funding here, and to check whether, and comparing those to see whether grantees would have more, would use their grant and their protective space more to go for novel research findings ex post. So what did we find? Well, first of all, in terms of selection, what we did find was that there was a bias against novel research here, but only for early career individuals. So for early career, the juniors, junior applicants here, the ones that had a record of having more novel research were less likely to be selected. And this holds even if they would be highly cited researchers here. So there was clearly a selection on highly cited. But if you were highly cited and at the same time also producing novel research, you were significantly less likely to be selected than other highly cited that would not produce highly novel research here. So clearly a bias against novel research here that's not mitigated by having a history of excellence here. So it's not just only the ones that do highly risky research that were biased against, but even if you were a highly cited researcher and you did combine that with a risky research profile, you were less likely to be selected here. And as we expected, the bias was stronger in the first stage here, but it was still also present in the second stage as well for the early careers. On the good side, there was no such penalty for risk-taking at the time of selection for the advanced researchers here. So it was really only the early career individuals that seemed not to be supported in terms of riskiness here, or were not allowed to do risky research in terms of likelihood for being selected as an ERC grantee here. So that's in terms of selection, and that was a bit of a surprise because ERC has this high gain, high risk mission here, but we, particularly for the early career individuals, still found that bias against novel research here. What did we find in terms of treatment? In general, we do not find any much significant effect in terms of treatment. So compared to counterfactuals that would be very similar, but without a grant, there was not really a big difference, which seems to suggest that all the grantees that are selected and continue to do good work afterwards here, it's really in the selection rather than in the treatment here. So they continue to do a lot of good high gain type of research here, but that's not because of the treatment, it's really because they are selected as being top researchers here. There was, however, one small exception where we did find a positive treatment effect for, again, these junior researchers, these starters, at least the starters in stage 2. So what we found was that starting grant recipients, compared to similar applicants that failed in stage 2, so that were almost selected, but eventually in stage 2 not, compared to these starting researchers here, those that did receive an ERC grant appeared to take on more novel research. But actually, if you looked a little bit deeper, what it actually meant was they did not reduce their novel research here, while those applicants, junior applicants that failed in stage 2, they actually cut back on their novelty research. And the ones that did receive the ERC grant, juniors, they did not do that. So the only positive treatment effect is that at least the starting grants that received an ERC grant were not cutting back on novel research here. They didn't have to cut back on novel research, while the ones that failed in stage 2 did cut back on their novel research. And we interpret this a bit as strategic behavior of those that failed in stage 2. They definitely want to get ready for reapplying next time here. And if they perceive that the ERC had this bias against novelty here, they were cutting back on that novel research in order to prepare better their profile for next applications here. And at least once you get a grant, you no longer have to behave strategically here and avoid the risky avenues here. So that's how we interpret those findings here. So overall, the ERC results, but also other results in the literature do seem to suggest that there is an issue of bias against risky research by funding agencies. So then the question is, why so what could explain this risk bias of funding agencies? And here we actually want to take a broad perspective and say that the bias against risk is something that's really a systemic problem. It holds in general in the research system. In general, a research system that has to prove its accountability here should be able to show results to taxpayers in relatively short time windows here. And that, of course, biases against taking too risky strategies here. So less tolerance of failure here. Another very strong characteristic embedded in the research system is this widespread use of bibliometrics to assess results. And as already indicated, these classic bibliometric indicators also inherently have this bias against risk here, because they go for the very quick results rather than the longer term results of the more risky research here. And of course, that's within the research system. That also means that universities that depend on the research system and on public funding, they will have to adapt to that by also being risk averse if they want to get sufficient funds from the research system here. So that's in general already a problem in the research system. If you go a bit more in terms of the research agencies, beyond that, why they would have a research a bias against risky research is because very often they are applying evaluation systems that are ill-equipped for funding risky proposals. First of all, because they take no portfolio approach, they just look at individual proposals, whether or not to fund or not, but they don't take an overall perspective of, are we sufficiently in our portfolio also allow for enough projects to be funded that are sufficiently risky here, like any venture capitalist would actually do here. So the way to manage risk is by a very careful portfolio analysis, but that's not what research agencies typically do here, by just only looking at proposals per individual proposal. If Very often they used field-specific panels and experts, and as we already saw, a lot of the risky research very often goes across specific fields here. And if you only have your own field experts, then again you run the risk that the big impact is not immediately seen here because that's typically seen outside your own field first. And then also the protocols that very often these research agencies use stress agreement here. And if you really want to go for agreement, these risky research very often leads to very polarized views. Some may like it, some may not like it here. And if you go for needs to be a consensus and an average, then you tend to go to averages where you miss these extreme scoring outliers on the two sides here. But then a very important point I also want to make in the whole system is that even if the research system would be willing to take risk, like for instance with the ERC here and the funding agencies, then very often they are also confronted with a risk aversion by principal investigators who may not be submitting risky proposals here. So not enough risky proposals. And this is, if we take a look at the rewards for doing risky research, there are rewards for doing risky research because there is this potential of breakthrough here, but it's typically insufficient to cover for the higher risk here. So the citation premium that you get for risky research is present, but is not large enough here. And then you get what we call the quad effect here, quad effect that we got actually from skating here. So if you look at female competitive figure skaters, how many points did they get for doing certain jumps here? The quadruple jump gets more points than the triple jump here, but the extra points it gets for the quadruple jump doesn't compensate enough for the higher risk that you take that it might fail here. And particularly the difference between the points for the quadruple versus the triple for females is way smaller than it is for males. And that's why females take less quadruple jumps than males here. So if there is not enough compensation in the system for the risky research, that in general builds in risk aversion by the researchers here. And if that's the case, even if agencies would be willing to go for risky research here, they might not get enough proposals in the first place here. So the research agencies already talked about here, and also this behavioral aggregation, how that leads to avoiding risk taking here. So overall, our conclusion from going through the literature on this of why there would be risk aversion is that this is really inherent in the system here. It's a systemic problem here, and it requires also addressing this systemic problem rather than just looking at individual programs of individual agencies here. But we do have, nevertheless, some suggestions to enhance risk-taking here. So first of all, try to de-emphasize the use of short-term bibliometrics here, and particularly the short-term characteristic that very often is used for bibliometric indicators like the journal impact factor here, because that really biases against the more longer term it takes for these higher risk breakthroughs to realize. Another possible solution or help to increase the risk taking is to increase the diversity of panel members. Make sure that you have panel members that are much more broader than specific fields, because they are also much better able to assess the broader impact of this risky research here. Allow for disagreement among panel members. For instance, experiment with wildcards, golden tickets here, so that it's allowed that certain panel members which have different views than others can still also be selecting their proposals as well. Take a portfolio approach. Also consider funding in stages here, such that the initial stages when they're still high risk here can be supported with smaller amounts of funding, and that you only gradually increase the amounts of funding when the research becomes later stage and with lower risk here. And that early stage funding is typically much more easy to be done at the local level here, at the research institute level here. So what we would advocate is also having these programs within research institutes where small amounts of money are used for funding very early on risky research here. And then finally, one other model I would like to discuss to close a bit my presentation and start the discussion, because there is a lot of discussion going on this, is should we need much more of a DARPA model here? Because that's what you typically hear. In order to incite way more risk-taking by funding agencies, we should use a DARPA model here. So, because DARPA is typically heralded as a model that's really the best in funding these high gain, high risk type of research here. Very often mission-oriented, high gain, high risk type of research here. Would that be a better model for to use here? Well, first we have to understand why it would be a success in terms of going for that high gain, high risk rewards. And we do have already quite a lot of research on that, trying to identify what the key success factors are of DARPA. And typically what the key success factors are is, first of all, the organizational flexibility here, an ability to really recruit program directors very flexibly. And these program directors are then giving quite a lot of autonomy to select really the projects that they think are really the high risk, high reward type of research here. And they can deviate from what's typically perceived by the peer community here. And that deviation is really very important here. So their autonomy to go for the really high risk, high gain type of in the hands of these program directors is very important here. Of course, they have to be able to be held accountable here, and they would have to have clear targets. But that goes together actually with being able to have a very clear mission-oriented program here, where these program officers also have very clear targets, but within these clear targets do have the autonomy to go for the high-risk profile here. So being able to attract those program directors here, and being able to give them autonomy while at the same time being able to help to hold them accountable, that's the success factor of DARPA here. And then the question is, and that's been applied for defense here, but can we also use that same model of DARPA beyond defense here? We now have discussion of ARPA for health, also ARPA for climate, and even a DARPA model for funding basic research here. But if you go back to the researchers that have looked at the key success factors of DARPA, what they say is actually it cannot be that easily transposed to other areas here. In order to have a DARPable domain here, that must be mission-oriented, mission-motivated here, where you can really identify much better what the target would be and identify KPIs for that. And so you need to really have these clearly identifiable goals and tractable progress metrics for which you can actually deploy your policy officers here. So that means that the DARPA model is actually not appropriate for funding basic research here, because there the mission of basic research is progress and knowledge. That's not sufficiently clearly identifiable here. So it's definitely not a general model for funding basic research here, because you cannot give that autonomy to these policy officers here, because it's too the progress KPIs that you need to give them are too unclear and cannot be focused enough. But that's not to say that you can use this DARPA model for very specific challenges in basic research here. Like if you go back to the mRNA example here, if you can really give a very clear mission in terms of solve the immune reaction to synthetic mRNA, that's a very clear mission here. For those kind of missions, you can use the DARPA model here. So overall, some very last slide here. I hope I've convinced you that bias against risk is an issue here, that it is important if we don't want to jeopardize these breakthroughs that would result from risky research here. But if we want to deal with it, given that this is multiplex and systemic here, we really have to work at the whole systemic policy response here. And what I think we definitely also need more is at least attention to this problem here, and also much more evidence and experiments on what could work and what could not work here. So I'm very much looking forward to hearing your suggestions and views on that, to see how we could push that agenda further here. So thanks for your attention here, and looking forward to your discussion.
Thank you so much, Renilde, for bringing us through the various aspects of risky research. I will now open the floor to the people following us online. As usual, you can use either the chat or the questions and answer options in Zoom, or if you want to talk, then please ask for that, and I guess that our technicians will be able to give you the microphone. Yeah, Marta Sanz wants to speak. I will unmute you. Marta.
Yeah.
Okay.
Can you hear me?
Yes,
Yep.
clearly, yes.
Yeah, thank you very much, Professor Feuerlers, for such an interesting talk. So my name is Marta Sansolé. I'm a mathematician. And well, I was very much interested in your findings on the ERC, because I have been a chair of the panel in mathematics of Consolidator, and so I know the system. But I was a little bit surprised about all the outcomes. So what you found, and my concrete question is whether the study you perform is global, or do you have results panel by panel?
Yeah. So we that's a good point. So we actually looked at all the life sciences and the physical sciences and engineering here. We excluded social sciences and humanities for the moment, because that was a bit more difficult to identify publications for all the panels here. We do correct for the fields and for the specific panels here. So we only compare within panels whether the ones that were more novel versus non-novel were biased against. But if we split by panel, so that we could also do to see whether there would be more bias for certain panels than for others. Of course, we couldn't go into too much detail here, but we did split into life sciences and physical sciences and engineering here. And actually what we found was that the bias against novelties is much stronger in the life sciences panels than in the physical sciences and engineering here. So there seems to be much less bias, which could also be perhaps explained by the fact that there is way more consensus on how to identify and codify what could be potential breakthroughs or not. Well, in life sciences, maybe there is more risk associated with novel combinations here. But I have no idea why. Otherwise, you could explain why in physical sciences and engineering there seems to be much less risk bias than in life sciences.
Yeah, thank you very much. And are there these documents available? Or
yeah, so when the slides will be made available, so yeah, there are the references to the papers that you can look at. Yeah.
Okay, thank you very much.
Yes, Marta, Professor Veugellers, before we open to the floor to the followers, so we were discussing for several minutes, and then she kindly offered to pass the presentation that we will get at the Institute. So if you are particularly interested, I guess, Renilde, that there is no problem to pass the presentation also to Professor Marta Sanz, right?
No, of course not. No. Yeah.
So,
thank you very much.
Okay. Thank you so much, Marta.
Okay.
Is there someone else who wants to ask a question or make a comment? Okay, if not, if you allow me, well, you have made at the beginning of your talk a very nice description of the history of the mRNA and also about the key players. Mm-hmm. Actually, I had the privilege to meet all of them. So Kariko and Weisman, and
Yeah.
also another one who played a very important role. His name is Robert S. Langer. That guy actually was a person who made possible, you know, to introduce the mRNA into the human body. Okay, so found the way, and actually the three of them, I met them because they were awarded the BBVA Agwar Frontiers of Knowledge to I think it was biology and biomedicine, no? And in the history you talk, you know, in particular about Langer. So he made a speech at the Agwar ceremony, which was enlightening, and it's perfectly aligned what the situation you explained it for Carico and Weisman. So he explained that during years and years, you know, he was knocking on all doors, you know, to explain that he had the potential to develop a technology allowing this to be real. And you know, he was fighted from everywhere, and you know, it's impressive. And at the end, you know, suddenly, well, the color of the situation changed dramatically, and then, well, he together with Carico and Weisman, they started to get funded and to get accepted everywhere and to get requested, you know, to submit proposals to give them more money and so on. So it's very impressive, no? And this brings me to another point, which is serendipity. Okay? Actually, I've heard some of these prestigious researchers from several places in the world, and some of them talk very seriously about serendipity. So they say, my only luck was that I had a boss, or I was working in an institution that allowed me to do the kind of research I wanted to do. So, and now we can maybe consider these risky research, but actually there is also this other component. What do you think about that?
No. I'm very happy that you raised it. So this, I guess, is Robert Langer. Was he from the Salk Institute?
Exactly.
Okay, yes. So I'm very much like the story that you raised here, and this issue of serendipity. I also believe in serendipity here. And I do believe that indeed there always are researchers enough in the system, like the Caricos or like the, and then also people willing to support them too. And like, for instance, the DARPA model also helped to support the first initial steps of the mRNA one here. So there are these bits and pieces here. So in the end, I do think we will always get the breakthroughs here. But the question still is, why should it rely on serendipity here? And could we not have a system that's way better organized? So it shouldn't be on these few individuals here, and that these few individuals, it's only if they are really persistent that they are successful here, and that there is a good match with. That's, I mean, we could make the system way better resilient for research here, so that we shouldn't be relying on this serendipity here, and that it should become way more mainstream here. That's my point.
Thank you. If no one else wants to ask or comment, then I will. Sorry. No questions in YouTube. Okay. Then I have a second. Sorry.
Yeah, Marta has raised her hand again.
Oh yes, Marta. Haha. Go ahead, Marta, please. The microphone is open.
Thank you. I believe in serendipity, but my question.
Me too. But we should.
I am a fan of serendipity. But well, my question is very, very concrete also. So, are there studies on the reasons for aversity of researchers to take risks? Because you have touched this question a little bit, but well, you went into another direction actually. And I think that in places like very conservative places, like, for example, I mean, or institutions in our country, unfortunately, there is this aversity from the side of the researchers, actually. And that's a serious problem.
Yeah.
So are there studies of the reasons for that, or?
No, that's a very good question. So there could be two things. First of all, researchers could have a preference for risk or not, right? And if they love enough risk, then I mean that's way more important than if they would be risk averse, right? Well, those kind of studies actually identify that researchers are a bit like entrepreneurs. They also have a higher preference for risk than on average here, than the average population here. But still, and that brings me to the next question, is if it's not necessarily that they would be risk averse researchers here, then it's a matter of, do they get the right incentives to take risk here? And I think that's the major story as to why researchers may not be taking enough risk here. It's not because they shy away from risk, but it's because the incentives, if you go in risky research direction, are not sufficiently compensating for the risk that you take here. So if you are rational then as a researcher, and you want to make sure that you have enough, that you can acquire enough funds, that you will get promotion, that you will get peer recognition, you will be punished against if you take too much risk here, and researchers will adjust to that here and therefore then take risk here. And then there is and then we'll avoid risk here, sorry. And if that's the case, then that explains more why they are risk averse than their risk preference or not. And that there is evidence in the system in the sense that indeed, this, like I said, like the citation premium, if you go for that novel research, there is a citation premium for getting higher breakthroughs here, but it's not sufficient to compensate for also the higher likelihood of failure here. So overall, the expected returns in terms of citation premiums from risky research are too low here compared to going for the more incremental ones here. So I think it's a matter of incentives rather than preferences.
Okay, yeah, thank you very much.
Thank you. You mentioned at the end of your talk, by the end, close to the end, DARPA.
Yeah.
So I have personally some experience of working with something similar to DARPA, which is ONR, the Office of Naval Research, which belongs to the US Navy. They also tend to fund, I would say risky projects. So quite close to DARPA.
Yeah.
And now you are perfectly aware that in the new framework program here in Europe, so we have the, these missions.
Yeah, right.
Have you analyzed the missions compared to DARPA? Do you see any similarities, or maybe originally the intention was close, but then the practical implementation is diverging? What's your view on that?
Yeah. Of course, the easy answer would be it's too early yet. But,
yes,
that's usually what they say. But so on the one hand, there is indeed it's much more mission oriented, and if it's more mission oriented, you can make it way more tied to really very clearly identifiable KPIs here that would allow for way more autonomy by policy officers to go for the high gain, high risk type of research here. So that part we might be getting more with that. The problem is that we don't have the organizational flexibility to recruit the program officers that have the capacity to then also select for and very closely follow also these projects in the next stages. And that's the part that's missing. It's the two parts. So it's the mission oriented, but it's also the flexibility to recruit the right type of people here. Like for instance, in Germany, they also discussed having this kind of mission-oriented DARPA type of problem here. But then, I mean, it got completely stuck in, they could only recruit bureaucrats. and that's not the right kind of people that you can give the autonomy with the mission statements here. So it's the two sides you need. And I think in the commission, it would be the same problem.
Yeah, I fully agree with you. And I would add also the reviewing system.
No, but the review system, and that's indeed because, like I already said, there is a lot of bias against risk in general in the system here because the review. And so if you rely too much on reviewers, then you get that risk. And then you need the policy officers, also in DARPA, they also use these reviewers here, but it's the policy officers that are able to compensate for the risk aversion by the experts in the field.
So thank you so much. I think that the people wants to, to have some dinner or something, because people have been living little by little, the remote followers. So again, Renilde, thank you so much for illustrating us about this interesting topic in your talk. Also, thank you, moltes mercès a tots els que ens heu estat seguint. And then I hope to see you next time in Barcelona. Hopefully.
Yes, me too. Yes, yeah. And then we
Thank
can
you so much.
discussion. Thank
Take
you.
care.